All right, welcome to the third video for our DNA and RNA module. So far, we went over the structure of DNA and RNA. We've discussed how DNA is replicated, so how we can copy and make new strands of DNA. Now we're going to talk about how you can go from a DNA molecule and take the information that that DNA molecule has and transcribe it into basically a different language, and that language is going to be that of an RNA molecule. And so basically, when we look at DNA, it really provides all of the instructions to a cell and an organism of what it's supposed to do. So, an organism's genotype, which is its genetic makeup, so the specific sequences of DNA that it has, is heritable information because that information is passed down from parent to offspring through their gametes because of meiosis. And that information is stored in those DNA strands, in those DNA molecules, which make up chromosomes. And the specific makeup, the genotype, ultimately determines what an organism will look like, what its physical traits are. So, again, what color is its hair? What color is the eyes? How tall is that organism going to be? Um, what is the feathers that are found on that organism? Are they flight feathers? So do they have pretty colors, etc.? So all of those physical traits are ultimately determined by its phenotype. And that phenotype is the expression of multiple proteins in the body. And those proteins are made based off of the instructions of DNA. So we basically have this pathway that the information travels from. And that pathway is DNA being transcribed into RNA. And then that RNA molecule being translated into proteins, into a specific polypeptide strand. And if you remember, proteins, which are one of our four organic molecules, are made up of multiple monomers of amino acids. And amino acids, we have 20 different ones that we use in building our proteins. And depending on the specific DNA sequence, a certain sequence of amino acids will be made, and that information flow has to go from DNA to RNA to that protein. So from transcription to translation. And so the function of the DNA gene, the specific sequences in that DNA molecule, determines what protein or polypeptide will be made. And so we have this relationship between genes and proteins that is a one-way information flow. And this is a little figure that kind of shows how that information flow happens. So again, we have DNA that's in the nucleus. It's transcribed into RNA, which also happens in the nucleus. That RNA molecule is modified and then leaves the nucleus via the nuclear pore and travels into the cytoplasm, where ribosomes will read that RNA molecule and build a protein based off of the information that's stored in that RNA molecule. Okay. And so, what exactly is the language then of nucleic acids, of DNA and RNA? Well, this language, specifically of DNA, is written as a linear sequence of nucleotide bases of A's, C's, T's, and G's. Now, a chromosome can be thousands to tens of thousands of nucleotides long, but if you remember, within an individual chromosome, you can have various sections that make up individual genes. So a gene is typically a few thousand base pairs or nucleotides long. And so what happens then is each individual gene is transcribed into its associated RNA molecule. So what you have is a segment of DNA being transcribed, and turned into an RNA molecule. And this process this process of going from DNA to RNA is called transcription. Basically, because we're going from a language of DNA being rewritten as a sequence of bases for RNA. So we're basically going from language of DNA to a language of RNA. Okay, and the sequence of nucleotides and the RNA molecule will directly determine the sequence of amino acids, which ultimately determines how your polypeptide will work and function, and the ultimate physical traits that an organism will have is the expression of those specific polypeptides and proteins. Okay? So there have been several experiments that have verified this flow of information from gene to protein. And this flow of information is actually based on what we call a triplet code. And this triplet code is made up of individual codons. Okay? And the instructions for this specific amino acid sequence in a polypeptide chain is really driven by these triplets or these codons. So basically, every 3 base pairs in DNA makes up a codon. And then that 3 base codon in DNA will be transcribed into a complementary 3 base codon in RNA. Then you have that codon in RNA that will be translated into amino acids that make up a polypeptide. And so this figure shows that process. So you have your DNA molecule, and let's say this green strand is a gene. It's a segment of our DNA molecule that codes for a specific protein. So a gene that codes for a specific protein that has a specific function. We look at that strand, and it has this specific sequence. So AAA, CC, GG, C, AAA. Okay? So now knowing the base pair associations, right? Remember, RNA has the same 3 base pairs as DNA, A, C, and G. The only difference is the T, which is now a U in RNA instead of DNA. And so G's still pair with C's, and A's instead will pair with U's in RNA. So for example, the transcription of this DNA strand will make an RNA strand that is UUU because U's pair with the A's, GG because these 2 G's pair with the C's, CC because they pair with the G's, G pairing with C, and then 4 U's pairing with those final 4 A's. So you end up with this specific RNA sequence after transcription. Now we look at the codons. And remember, the codons are the triplet base pairs. So every 3 base pairs make up a codon. So UUU is one codon. GGC is another codon. CGU is another one. And UUU is the last codon. And each of these individual codons specify a specific amino acid that should be in that sequence of amino acids that make up the polypeptide. So basically here, the codon triple U says you should have a square. GGC says it should be a sphere. CGU is a triangle. And again, we have triple U, so we have another square. And so this specific sequence of amino acids is driven by this codon, these triplets, which is in turn driven by the original DNA strand. So you have that transcription to translation, which goes from DNA to RNA to polypeptide. Okay. And so now we have this genetic code. Right? The genetic code, again, remember, is that set of rules to convert nucleotide to RNA to an amino acid. Out of our genetic code, so our 4 base pairs, if you have 4 base pairs and you look at every combination of 3 base pairs out of those 4 to make your codons, you end up with 64 possible triplets. So 64 possible codons. 61 of those combinations code for 1 out of 20 amino acids. 3 of those triplets, those codons, are what we call stop codons. It basically tells the machinery to stop making the polypeptide. So you actually have a start codon, codons in between the start codon and the stop codon. And so the start codon tells a protein to start being made, and a stop codon tells a protein to stop being made. And a given RNA triplet always specifies a given amino acid. So for example, AAU will always specify for the same amino acid. GCA will always specify for its associated amino acid. And researchers have went in and looked at which codon specifies for a specific amino acid. Okay? And so before we move on, let's just think about this question here. How many nucleotides are necessary to code for a polypeptide that is 100 amino acids long? Okay? So how many nucleotides do you need to make a protein, a polypeptide, that contains 100 amino acids? So take a minute and think about this. All right, so if we have 100 amino acids, every amino acid has to have a codon, a triplet associated with it. So one amino acid is 3 nucleotides. So if you have 100 amino acids, that's 100 times 3. So you need 300 nucleotides in order to generate 100 amino acids. Okay? So this table here is how you can determine what amino acid is determined by a specific codon. So with this one, you start in the middle and then you work your way out. So for example, let's determine what amino acid is made by the codon AU, come on, AUG, okay? AUG. So pause the video, take a minute, and try to figure that out. All right, so hopefully you were able to deduce that it is the start codon, methionine. So first what you would do is start in the middle, you would find A. Okay, so here's A. Next, you would find the second codon, which is U, which is right here. Then you would find the third codon, which is G, which is right here. So basically you say A, U, G, and that tells us it's methionine. Okay. All right. So let's do another. Let's do G, A, C. Okay. So take a minute, see which amino acid that codes for. All right. So hopefully you found this. So G, A, C. So aspartic acid is the amino acid that it would code for. Okay. And one thing I also want to point out is that there is a considerable amount of redundancy in these codons. Okay, so what I want you to notice is if you have a codon that is, let's say, A, Actually, let's do this. Let's say you have a codon that is A C blank, right? The amino acid that's going to be made by AC, and you can put in any one of these four, right? G, A, or U, or C, is going to be threonine, right? See that? A, C, and then if it's ACG, it's threonine. If it's ACU, it's threonine. If it's ACC, it's threonine. If it's ACA, it's threonine. So there's some redundancy there. Okay. So what that means is, if you have a codon that's ACG in your DNA, let's say a mistake happens in the copying process, and instead of ACG, we now have ACA. Even though the codon changed, the resulting amino acid did not change. That means that you can have some mistakes, but the protein will still work and function like it's supposed to. So generally at the third codon, you can get mutations that aren't necessarily detrimental. They don't really change anything. However, if you change the second and especially the first, you're most likely going to get a change in the amino acid. So for example, if we go from ACG to GCG, we've completely starting in a whole different quadrant of the circle. So now it's GCG, we're at alanine where we would have been at threonine, right? Same thing for the second codon. So let's say we go from ACG to AGG, right? So now instead of going AC, we're now going AG, and now we're at arginine, a completely different amino acid. And the reason why I'm saying this is just to say that there is redundancy in the code to help minimize these detrimental changes. But these changes can happen and completely change and alter the functioning and whether or not the protein works. And what we also can see here is that methionine is the only start codon. So every protein will start with methionine. So it will start with AUG, and then it will have all of these codons in between, and then the final codon will be one of these three stop codons. So for example, this one is UGA, or maybe it's UAA or UAG. So those three stop codons would be at the end. So let's say we do UGA, right? So you would have your start, you would have the interior, and then you would have the stop. Okay? And so that's how the codons and the triplet works in order to code for specific protein and polypeptide sequences. What's really interesting with the genetic code is that it's almost nearly universal. What this means is that it's shared by organisms from all different kinds of evolutionary levels, from the simplest bacteria to complex plants and animals and vertebrates. We all share DNA. And there are specific sequences that are similar in our DNA from bacteria to humans. We have these shared sequences that are the same. And that suggests that shared evolutionary history with all living organisms. And so the universality of this genetic vocabulary really tends to hold a lot of evidence for the fact that very early in evolution, the genetic vocabulary arose and was basically passed on to all other living organisms today. And because we all share this common genetic code, it's possible to program one species to produce a protein from another species. And so scientists have actually done studies where they mixed and matched genes from various species. So for example, this is a bioluminescence gene that was implanted in I believe this is a mouse, to where its forelimbs are fluorescent. And that gene came from another organism. And so because of that universality, this can happen. Okay, this is possible. All right, so now finally getting into the details of transcription. Okay, so going from DNA to RNA. So transcription, like we mentioned, is transferring of genetic information from DNA to RNA. So when you have DNA replication, you have two strands that separate, and then that strand is replicated. Okay. Transcription is a little different from where replication uses both strands. Transcription only uses one of the DNA strands as a template for the RNA molecule that's going to be made, and the other strand is not used. Okay. The nucleotides that make up the new RNA molecule are put into place one at a time along that strand as the enzyme, the RNA polymerase, goes in and forms hydrogen bonds with the basis of that template DNA strand. And RNA nucleotides also follow those base pair rules like we mentioned a couple slides ago. The only difference is that U is there instead of T. So U pairs with A and G still pairs with C. And then again, those nucleotides are linked together through that RNA polymerase. So that RNA polymerase is building that RNA molecule. Okay. So with transcription, you have initiation, elongation, and termination. So those are the three steps of transcription. So what happens is there's a promoter region at the beginning of the DNA sequence that basically tells the enzymes to start transcribing here. Okay, and so they will start at that promoter region and start transcribing the RNA polymerase. And so that promoter area is where the RNA polymerase attaches to the DNA sequence to start that transcription process. So that first phase again is called initiation. That's where that attachment happens of the polymerase. During the second phase, which is elongation, that's where all of your individual bases are added to the RNA molecule and the strand just grows longer. And as that strand lengthens, it peels away from the DNA template, causing the separated DNA strands to come back together. So basically the DNA strands will be split. One strand will be used as a template, and as that strand has been used, the region behind that has already been used will close back up. That way there's not a large portion of the DNA molecule that's separated at any given time. After this process of elongation, termination happens. Okay? So the third phase, which is termination, is where the polymerase reaches the terminator, which is signaling, okay, here is the end of the gene. This is where you stop transcribing the RNA molecule. And then once the polymerase detaches, the DNA strands completely rejoin together. Okay. So here's a figure that just kind of shows this process. So you have your RNA polymerase, you have your DNA molecule, which is the blue double helix. That DNA molecule is split, and the individual nucleotides are attached. And as the polymerase continues along the strand, like we see in the second and third figure, the DNA helix will close back up. Right? So there's only a small portion of the DNA helix that's separated at any given time. And we also see that that initiation process is where that polymerase binds to that promoter region. You have that elongation. And then when it reaches the termination end, then the RNA polymerase will detach. The DNA molecule will close back up, and then you will end up with a completed RNA molecule. Once that RNA molecule is made, it then has to be modified a little bit in eukaryotic organisms. In prokaryotic organisms, that transcribed RNA is immediately functional. It's immediately functional as what we call mRNA or messenger RNA. And messenger RNA is the molecule that's translated into a protein. However, the eukaryotic cell, like I said, needs to be modified a little bit before it's recognized as an mRNA molecule. It first has to have what's called a cap and a tail added to the ends of the mRNA molecule. And then it also has to have some of the molecule removed from it. And so basically, within our DNA strands, there are, even within a gene, you have some portions of that gene that don't really code for anything, okay? And it doesn't have really any information. And those non-coding regions are called introns. The coding regions, which are important for telling the ribosome what kind of amino acid to attach to the growing polypeptide, are called the exons. Okay? So after that first RNA molecule is transcribed from DNA, it gets the cap and the tail put on. Then an enzyme will go in and it will cut out those non-coding regions. So basically, you think of a ribbon that is, let's say, two different colors. You have blue sections and purple sections. Let's say the introns are the purple section. So you'll have that ribbon, you'll take the scissors and you'll cut out those purple sections. And then you'll tape all of the blue sections, the coding regions together, the exons. So your entire RNA molecule is now made up of only exons, okay? And that process is called RNA splicing, okay? So both of those introns and those exons and introns are transcribed from the DNA into the RNA. But before the RNA becomes fully functional mRNA, that RNA splicing has to happen. And it is a very important process. And RNA splicing is actually thought to be really important for humans in allowing our 21,000 genes to produce several times the number of polypeptides, because you can vary the exon combination in the final mRNA structure and end up with a completely different protein, right? So depending on how you cut out and splice those exons together, you can end up with different proteins. So here we have a figure showing kind of this process. So you have your DNA, you have your exon, which is your coding areas, and your intron, which is your non-coding areas. When you translate that, they still exist in that RNA molecule. Once that RNA molecule is made, then the cap and the tail is put on. Those are the yellow areas. Then the RNA splicing happens. Those introns are cut out, so those pink sections are cut out and removed. The red sections, the exons, are spliced together. Now, this is the important coding sequence. This is what's going to tell and determine the specific polypeptide arrangement and make up the protein or amino acid arrangement. Once the splicing has happened, you then have your messenger RNA that then leaves the nucleus via a nuclear pore and can then be translated into a protein. And so that's it for this video on translation. We'll pick up next in our next video looking at, I'm sorry, not translation. We just talked about transcription. We'll pick up in our next video focusing on translation. So how RNA is translated into a functional working protein. Okay? And we will see you in the next video.